Hi guys, this is STAAR Day 9 on page 118. This video is just basically some reminders to help you for your tests. Some things I wanted to say before you guys go in. The first thing I'm going to talk about is going to be photosynthesis and cellular respiration. Basically all I wanted to remind you of there is when we look at photosynthesis, remember all of the things you have to give a plant in order to survive. So if I were to look at my plant, the stuff a plant absolutely needs is they absolutely need H2O. They absolutely need solar energy. And then you might not think about it, but plants also need to have carbon dioxide coming in through their stoma, through their leaves, to help them with photosynthesis. So what's actually happening when they're doing photosynthesis? Inside of the cells, they have these little organelles. Because remember, an organelle is something small. It's a small working part inside of the cell. So they've got these organelles called chloroplasts. They take in all of the three things I listed, solar energy, carbon dioxide, water, and they put out C6H12O6, which we know is going to be glucose, and then they also give out O2 gas, oxygen gas. The opposite, so as long as you remember this equation, you're going to be good. The opposite would be what happens in the mitochondria. Remember, mitochondria is always going to look really squiggly like that. The mitochondria takes these things. So whenever we eat plants or whenever we eat food, we're taking in glucose. Whenever we breathe, we're breathing in oxygen gas, and we're using it in our mitochondria. The reason why is we're putting out water, we're putting out carbon dioxide, and we're putting out ATP. So notice one of the differences between cellular respiration and photosynthesis is cellular respiration is putting out energy, photosynthesis is putting in energy. Okay, so as long as you remember one of the equations, you'll be good to go. You should know the opposite of it. So as long as you remember you have to give a plant water, you have to give a plant carbon dioxide, you have to give a plant solar energy, you can remember that we put out glucose, put out oxygen, and then for cellular respiration we do the exact opposite. We put in glucose, put in oxygen, give out H2O, carbon dioxide, and ATP. Alrighty, just something else I wanted to remind you of for cellular respiration and photosynthesis. Please don't forget that plants, just because they're doing photosynthesis, doesn't mean they can actually use the energy from the sun to power things inside of their body. They need ATP just like we need ATP, which means they also do cellular respiration. Okay, so plants, animals, fungi, everything you can think of has to do cellular respiration. Okay. Alright, the next topic is going to be mitosis and meiosis, and we're going to cover also a couple of things as we go through this. Don't forget that mitosis is part of the cell cycle. Okay, and then we've got meiosis on the other side. So for the cell cycle, when we're looking at it, if this is my original cell, I'm going to get two cells out at the end of the cell cycle. If my original cell had one chromosome from mom and one chromosome from dad, so these are identical chromosomes, just one of them came from mom, one of them came from dad, my cell at the end will be identical also. So both of these cells will have the exact same stuff. These cells are going to be diploid, di meaning two. So if here my number was 2, here these will also have 2 chromosomes. Okay, so if you see a diploid cell, if it has a mom and dad copy of the same chromosome, diploid immediately. Okay, meiosis on the other hand, if this is my cell originally, I'm going to go through cell division twice. So notice how this is cell division 1, this is meiosis 1, and now I'm going to go through meiosis again and divide one more time. So I'm going to get 4 cells out of this. And if my original cell had a purple one and it had a blue one from mom, one from dad, basically what I do is I copy these over so they get a little bit larger. That's where we get the X shape. And then I'm going to end up splitting them. So this cell right here will get those, and this cell right here will get these. Now actually I just simplified the process a little bit. We do remember the process of crossing over. So for crossing over, actually that leg of the chromosome should go here. So I took a chromosome leg from here and I swapped it with here. So actually process gets a little bit more complicated because I actually do the swapping of chromosomes. That's going to give me variety in all of my kids. Okay, so if we look right here, that's actually how it should look. All of my cells, all of my kids' cells, they're going to be gametes, and so sperm or egg, and they are all haploid. Remember, haploid means half. So if here my original number was 2, here I will half that number. They will only have 1 chromosome in each. So all of the kids' cells only have 1. Sorry, I say kids, but I actually mean sperm or egg. Alrighty, so that was mitosis and meiosis. Another thing we need to talk about for mitosis is what are the actual phases. Remember, we start with interphase, and that's when I have one nucleus and I'm doing gap 1, S, and gap 2. Then we go to prophase. Prophase, my nucleus breaks apart, my chromosomes crisscross. We go to metaphase, my chromosomes line up in the middle. Notice my hands are not clasped, I have no nucleus. Then I go to anaphase, my chromosomes get pulled apart. Telophase, I get 2 nuclei, and cytokinesis, they separate. The way to remember that is going to be, I pretend my aunt's turtle cries. So this one right here, I pretend my aunt's turtle cry. That's the actual stages of mitosis. Meiosis uses those same stages, prophase, metaphase, anaphase, telophase, cytokinesis, but they do it twice. Okay, so both cell cycle and meiosis use these phases. Make sure you know the names, make sure you know what's happening. Cell cycle only does this once, meiosis does that twice. Okay, so they do cytokinesis 2 times. Alrighty, so we talked about that one. Good. The other thing is, what is the purpose of the cell cycle versus the purpose of meiosis? Well, mitosis, the cell cycle, its job is for growth and repair. So what we say is mito is growing. Mitosis is for growth and repair. And remember, mitosis is a part of the cell cycle. Okay. The next thing I wanted to talk about for mitosis meiosis is cancer. Cancer is basically when you go through the cell cycle over and over and over again. So if I were to look at that, here's my original cell that I started with. It went through the cell cycle once, so I got 2 cells. Awesome. If this is cancer, I'm going to go through the cell cycle again, and now I'm going to have 4 cells. I'm going to go through the cell cycle again. I'm going to get 8 cells. Okay, and then I'm going to go through it again. I'm going to get 16. I'm going to go through it again and again and again and again. Now I have an entire little clump of cells that I never wanted. This clump of cells is going to be called a tumor, and we know that tumors are associated with cancer. So if I go through the cell cycle an unhealthy amount, meaning it's unregulated, I can't control it, I can't stop it, this is cancer. Alrighty, so now we talked about cancer. All right, perfect. Let's go ahead and move on to another topic. The next one is going to be for Punnett squares and then like sex-linked inheritance. So all I wanted to remind you of there when we talk about complete, incomplete, let's say for example that I'm talking about flower color. And you can get, so we're talking about the gene that tells me to make flower color. Remember the word gene means a group of DNA that tells me how to make a protein. So if I'm looking at the gene that tells me how to make the pigment for flower color or the protein for flower color, you can either get a dominant R, which gives you red, or you can get a recessive, which gives you whites. All right, so for this gene, here are my 2 options. These are my 2 alleles. You can either get the red version or the white version. Well, let's talk about sperm and egg. So if this is my ugly looking sperm. So if these are my sperm and egg and they will combine to make you a zygote, remember these are haploid, and this cell is a diploid cell. If, let's say for example, mom had a dominant R, dad had a lower recessive R. Well, when they come together to form you, you are going to be heterozygous. Okay, so let's look at what these Punnett squares would look like. If we're talking about complete dominance, meaning a regular type question, for you, you're heterozygous, what color will you be? Your phenotype will be red because you have 1 capital R. If this is codominance, and it'll tell you in the question if it's codominance, if you're heterozygous, I will see both because co always means both. So red and white, you will be spotted or you will be striped, for example. Okay, but you will see both at the same time. If this is incomplete, remember incomplete means a mixture. So if you are heterozygous, I'm going to mix red, I'm going to mix white, I'm going to get pink. Okay, so just remember that. Alrighty, the next one is Punnett squares. Why do we write Punnett squares the way we do? Just remember that you have to split the letters. So if we're doing a 4-box Punnett square, if this is my parent, okay, so one parent looks like this, one parent looks like this. For this parent, I split the letters. For this parent, I split the letters. If we're doing a 16-box Punnett square, and let's say here's one of my parents, and here's my other parent. Okay, when I do my big 16 box and I do all of my parents right here, I do FOIL, and so this R can go with capital B. It could also go with little b. I'm just basically getting all of my options of R and B. And then I go over here, that little r can go with a capital B, little r can go with little b. And then I do the same thing for this parent, and I'm done. Okay, so just remember how to do that. Let's see. The next thing I wanted to remind you of is going to be like active and passive transport. So remember, we draw the hill. Please draw the hill. Here's the top of the hill. Here's the bottom of the hill. If I'm pedaling up a hill, I have to do active transport because I need ATP. If I'm at the top of the hill and I'm going down, it's going to be passive transport. No ATP. And I never write correctly when I do this, okay? So this is passive, that's active. The other thing I want to remind you of is when we talk about osmosis. Osmosis is the movement of water, not sugar, not salt. If I'm talking about active or passive transport, I'm talking about moving salt or sugar. If I say osmosis, I'm moving just water. And remember, there are 3 times I move water. If I say hypo, that means a number is below. That means the amount of salt or sugar on the outside is low, so it might be like 1% on the outside and like 10% on the inside. So this number is below, water moves in. If I say hypertonic, hyper means above. And so if I say above, that means this number will be 10, this number will be 1, water moves out. And then of course, if I say iso, iso means equal. And so for isotonic, water moves in, water moves out because the numbers are the same. The next thing I wanted to remind you of is going to be Dear King Philip. So remember when we do the taxonomy or the taxa, it goes in order of domain, kingdom, phylum, class, order, family, genus, and species. Another way to remember that is Dear King Philip came over for great spaghetti or soup or whatever words you want to use. I don't care what the words that you use as long as you remember the correct order. So Dear King Philip came over for great spaghetti helps you to remember it so you can write it. If you don't remember the song or whatever it is, you can always write it down. Dear King Philip came over, okay? Please remember that this is the most general and this level is the most specific. So because it is the most specific down here at the bottom, if you make it in the same domain, same kingdom, same phylum, class, order, family, and you make it to the same genus also, you and that other organism are very closely related. However, if you only have the same domain and the same kingdom, but you have different phylum, class, order, family, genus, you're not very closely related. Alrighty. One other thing I wanted to remind you of is whenever we write scientific names. So let's say, for example, my scientific name is Felis catus. This is my scientific name. That letter, the first word should be capitalized and it should be underlined or italicized. The first word right here, so Felis, is going to be the genus. The second word right here is always going to be the species. Let's say, for example, I'm looking at Felis catus and Felis concolor. Okay, so if I'm talking about Felis catus and Felis concolor, again, Felis is the genus, concolor is the species. Notice they're in the same genus together. That means they have the same domain, same kingdom, phylum, class, order, family, and genus. So they have all of those levels in common. Because they have so many levels in common, I know that those two are really related, but they have a different species. Because they have a different species, I know that they're 2 different types of animals, 2 different species of animal. Alrighty. What else do I need to remind you of? The structure of an amino acid. For when we talk about an amino acid, it'll always have a carbon bonded to a carbon, and then this carbon will always be bonded to a nitrogen. So when we talk about what elements we find in the biomolecules, proteins, because remember amino acids are amino acids are building blocks of proteins, they will have a nitrogen, carbon, oxygens, and then they will have hydrogens inside of them. I just wanted to throw that out there so you can remember that. Let's see, define a population. Remember, we go biomolecule. From biomolecule, we say a bunch of biomolecules make up a cell. A bunch of cells make up an organ. A bunch of organs make up an organ system. A bunch of organ systems make up an organism. A bunch of the same organism, a bunch of the same species living in one area is the definition of a population. So if we just looked at Samuel and we just looked at the humans living in Samuel, we are in one area, we're all in Samuel, and then we're all human, we're the same species. This is a population. But I know obviously Samuel has way more than just humans in it. It has animals, it has the dogs that walk the streets, it has plants, it has all these other things. So we are actually a community, a bunch of populations living in one area. That's a community. And then a bunch of communities make up an ecosystem, and then a bunch of ecosystems will make up the biosphere, so all of Earth. Please make sure you know the definition of an organism. Make sure, oh, I skipped tissue. A bunch of cells don't make an organ, they actually make a tissue, and then a bunch of tissues make up an organ. Forgot to put that in there. Alrighty, so please make sure you know the definition. Please make sure you know what you're looking for. They might give you a question that says, kidney cell is the same level as, and you have to know that if you're looking at a cell, you're looking for another cell in your answer choices. Okay, so please make sure you know what you're looking for. The last things that I wanted to remind you of are enzymes. Please make sure that you know that enzymes are actually proteins, and their job is to speed up a chemical reaction. I'm going to write chem reaction. How are they speeding up a chemical reaction? Well, if this is how much energy I need without an enzyme, this is my amount of activation energy. With an enzyme, it's going to be much lower. Okay, so notice what they did to my amount of energy I needed. Okay, so they speed up a chemical reaction by lowering activation energy. Oops. Okay, the next topic is going to be nitrogen. For nitrogen, remember nitrogen makes up amino acids, and amino acids make protein. So you need nitrogen to make those amino acids, and we also need nitrogen to make up DNA or nucleic acids. So it's very important for that. The other thing about nitrogen is, how does nitrogen get in the soil? Nitrogen gets in the soil two ways. One, when you get when you decompose and you die. The second way is actually when you poop. Your feces, your defecation has a lot of nitrogen inside of it. So those are two ways nitrogen gets into the soil. And then remember, nitrogen fixing bacteria will take nitrogen from the air and put it in the soil. And then bacteria will also break down the poop in the dead animals. So bacteria is really good because it's breaking down or it's taking nitrogen from the air. The last thing you need to know is the carrying capacity. Carrying capacity is basically the highest number of organisms that can survive in an area. So for example, if here's 26, if this represents my classroom and this represents the number of seats, okay, so if this represents the number of chairs, if this represents the number of chairs, the max number of individuals that I can keep in my classroom are going to be 26 people, because that's the maximum number of chairs. So see right here how it flattens out? This is my carrying capacity. So if you're looking for the carrying capacity, you're looking for the flat part of the graph that goes long ways, because that's the highest number I can keep alive. All right, good luck.